Inside every single cell of your body, right now, a molecule is being read, copied, and translated into you. Your eye color, your blood group, your height, all encoded in just 4 letters. Welcome to day 27 of Biology Domination. Today, we unlock the molecular basis of inheritance. This single chapter fetches 5 to 6 marks in NEET every year. So lock in, because by the end of this video, DNA replication, transcription, translation, and the Lac Operon will feel like second nature. Before we could study inheritance at the molecular level, one question had to be answered. Is it DNA or protein that carries genetic information? Griffith, in 1928, showed that something from heat-killed smooth bacteria could transform live or rough bacteria into virulent smooth ones. He called it the transforming principle. Avery, McLeod, and McCarty later confirmed it was DNA. Then, Hershey and Chase in 1952 used radioactive tracers, P32 on DNA, S35 on protein, and showed that only DNA entered the bacteria when a bacteriophage infected it. Case closed. DNA is the genetic material. But, and NEET loves this trap, in organisms like TMV, tobacco mosaic virus, HIV, and QB phage, the genetic material is RNA, not DNA. Write that down right now. Now, let's build DNA from scratch. Watson and Crick gave us the double helix model in 1953, Nobel Prize-winning work. DNA is a right-handed double helix. The 2 strands are anti-parallel. One runs 5' to 3', the other runs 3' to 5'. The sugar phosphate backbone is on the outside. Nitrogenous bases are inside, held together by hydrogen bonds. Adenine pairs with thymine, 2 hydrogen bonds. Guanine pairs with cytosine, 3 hydrogen bonds. Chargaff's rule, A equals T, G equals C. For measurements, the pitch is 3.4 nanometers, the rise per base pair is 0.34 nanometers, and there are exactly 10 base pairs per turn. Purines, adenine and guanine, have double ring structures. Pyrimidines, cytosine, thymine, and uracil, single ring. These numbers appear in NEET almost every year. Screenshot this slide. DNA replication is semi-conservative, proven by Messelson and Stahl in 1958 using heavy nitrogen N15. Each new DNA molecule contains 1 original strand and 1 newly synthesized strand. Replication begins at the origin of replication. In bacteria, like E. coli, there is only 1 origin, but eukaryotic cells have multiple origins to speed things up. At the replication fork, helicase unwinds the double helix. SSB proteins keep the single strands stable. Primase adds an RNA primer because DNA polymerase cannot start from scratch. It can only extend. Then, DNA polymerase III synthesizes the new strand always in the 5' to 3' direction. On the leading strand, synthesis is continuous. On the lagging strand, synthesis is discontinuous. It happens in short fragments called Okazaki fragments. DNA polymerase I removes the RNA primers, and ligase seals the gaps between Okazaki fragments. NEET loves to ask, which enzyme joins Okazaki fragments? The answer is ligase. Transcription is the process of making RNA from DNA. The template strand runs 3' to 5', and RNA polymerase reads it in this direction to produce mRNA in the 5' to 3' direction. Unlike DNA replication, transcription does not need a primer. RNA polymerase binds to the promoter region and begins synthesis. In bacteria, 1 RNA polymerase does everything. In eukaryotes, RNA polymerase I makes rRNA, RNA polymerase II makes mRNA, and RNA polymerase III makes tRNA. In eukaryotes, the primary transcript is called heterogeneous nuclear RNA, or hnRNA, and it needs to be processed before it becomes mature mRNA. This processing involves 3 steps: splicing, cutting out the introns and joining the exons; 5' capping, adding a modified guanosine cap; and 3' polyadenylation, adding around 200 to 300 adenine nucleotides as a poly-A tail. These modifications protect the mRNA and help in its export from the nucleus. Translation is the process of decoding mRNA to build a protein. This is the final step of the central dogma: DNA to RNA to protein. The genetic code is a triplet code. 3 nucleotides called a codon specify 1 amino acid. With 4 bases, you get 64 possible codons. 3 of these are stop codons: UAA, UGA, and UGS. The remaining 61 codons specify amino acids. AUG is the start codon. It codes for methionine, and it's the only codon for methionine. The genetic code is degenerate, meaning multiple codons can code for the same amino acid. It is also nearly universal. The same codons specify the same amino acids in almost all organisms. Translation happens at the ribosome. The ribosome has 3 sites: A site, where the new amino acyl tRNA enters; P site, which holds the growing polypeptide chain; and E site, where the empty tRNA exits. In prokaryotes, the ribosome is 70S, 50S plus 30S. In eukaryotes, it's 80S, 60S plus 40S. The Lac Operon is one of the most important concepts in this chapter, and NEET loves it. Proposed by Jacob and Monod in 1961, the Lac Operon is a classic example of gene regulation in prokaryotes. It controls the metabolism of lactose in E. coli. The operon has a regulator gene, a promoter, an operator, and 3 structural genes: LAC-Z, which makes beta-galactosidase; LAC-Y, which makes permease; and LAC-A, which makes transacetylase. When lactose is absent, the repressor protein is active. It binds to the operator and blocks RNA polymerase from transcribing the structural genes. When lactose is present in the medium, it gets converted to allolactose, the true inducer. Allolactose binds to the repressor, changes its shape, and the repressor falls off the operator. RNA polymerase can now transcribe the structural genes, making the enzymes needed to digest lactose. This is called negative regulation. The operon switches off when lactose is gone. NEET trap: the Lac Operon is inducible. The trip operon is repressible. Know the difference. Let's do a rapid-fire recap before you go. The central dogma: DNA to RNA to protein. DNA replication is semi-conservative, proven by Messelson and Stahl. RNA polymerase needs no primer, unlike DNA polymerase. AUG is the start codon. UAA, UAG, and UGA are stop codons. Prokaryote ribosomes are 70S. Eukaryote ribosomes are 80S. The Lac Operon is inducible. The trip operon is repressible. RNA is genetic material in TMV, HIV, and QB phage. NEET traps: Okazaki fragments are only on the lagging strand. DNA pol 1 removes the RNA primer, not pol 3. GC pairs have 3 hydrogen bonds, stronger than AT with 2. And the actual inducer in the Lac Operon is allolactose, not lactose itself. Download the PDF notes from the link below. Day 28, evolution drops next. See you there.